Yeah.
It's Monday morning, you tired? Well, welcome to a dreary Monday morning. This week when you come into lab, you will be asked to turn in your lab questions from last week at the beginning of the period. The reason we want them at the beginning of the period is we don't want you in there trying to write out answers to your questions during the lab, so they're due at the beginning of lab. I also want to reiterate something that was in the instructions for the lab handout, and sometimes this becomes something that students just really aren't aware of. You have a lab partner to help you many times gather data, to work with, and also to talk to about what's going on, even someone you can talk through answers to lab questions and things like that. But when it comes to handing in something, it has got to be your own work. It has got to be your own words. You cannot just take someone else's lab handout, even though they were your partner, and just copy down exactly word for word what they said on their answers. Okay? That is academic dishonesty. And so please be aware of that. It's fine to talk to people about the concepts, but when it comes time to writing your answers, you really need to write your own answers. Okay? Okay, how many of you feel like you are reasonably competent working with a microscope? Okay, a lot of you do. Even those of you who say you feel reasonably competent now are probably making some mistakes in using the microscope. So this morning we want to talk about kind of a systematic way you should go about using the microscope, not only in this lab, but in any biology lab that you'll have from here on out. The purpose of this lab is twofold, really threefold, I guess. One is to get you comfortable using the microscope. This is the only microscopy lab we will have in this course, but you will have microscopy labs in other courses. If you make it to your senior year and still are interested in biology, you might take microbiology, and all of the labs in micro use the microscope extensively. Second purpose of this is to get you to look at some cells. I think it really helps to make cells more relevant if you look at living cells rather than pictures or even videos of cells. Take your time today and just really kind of take it all in and don't rush through it, because especially when we look at some of the living things, they really are pretty fascinating. Spend some time with them. And then the third thing we're going to do is we're going to kind of take you through a little kind of evolutionary history from prokaryotic cells to single-celled eukaryotes, then some multicellular, simple eukaryotic organisms, and then end up by looking at some slides of tissues of epithelial tissue, such as that you have in your gut. So hopefully you get an idea of the evolution of cell complexity as a result of this. Okay, these are the microscopes that we're going to be using in lab. This is our Olympus CH30. They're already set out for you. If you lift them up, they're very hefty. And if you ever have occasion to carry those microscopes, like putting them back in the cabinet where they come from, always make sure you grab them by two hands. Put one hand on the base. There's actually a little kind of handhold in the back of the base, and then the other one underneath right here. They're really hefty. Don't drop them. They're expensive. They're, $1,100, $1,200 a piece, and so we want to make sure that they stay in good shape. Here are some of the features that you need to be aware of. Some simple things like, where is the on-off switch? Obviously, the cord has got to be plugged into a power supply first before you can turn it on and off. That happens. This adjusts the intensity of the substage illuminator. And so when you flip this on, then if you turn this clockwise, you'll get more voltage going through this little substage illuminator, and the light will begin to shine up through this device called a condenser, and eventually through the stage. Your slide will be sitting on the stage, and then one of 4 objective lenses will be in place, and the light will come up through the specimen, through the objective, and then eventually out and through the oculars. Okay? What is the relationship between the image that you see and what's actually on the slide? Is it exactly the same orientation? What's the orientation of what you see compared to the real image?
Is it
It's inverted, it's upside down, and it's also backwards. That can create a few problems when you are trying to look and find something on the microscope slide, because it's kind of counterintuitive which way you move the slide to see things. So if you want to see something at the top, you have to move the stage actually away. The stage is where you put your slides. There is a little clip system right in here that's intended to hold that stage in place, that slide in place. There is a little lever right here you can use that will swing this little arm back, and then you just nestle the slide right in here, and the arm retracts again and holds that slide in place. Don't just set them on top of this metal stage, and don't try to jam it underneath there, okay? Flip this little lever, and then it will just nestle right into this little square right here, and in that way that will be held in place. This is your movement dial for the stage, and it moves both in the X and the Y axis, and there are two different knobs here. You can turn one to move in X and one to move in Y. It's not a bad idea when you get your first slide to just kind of play around with it a little bit and get comfortable with this optical inversion phenomena. That means you've got to move things the opposite direction for the way you want to see the image move. So that's important. The condenser you probably won't have a lot of occasion to work with today. I'll show you on another slide, but there's a little focus knob that allows you to raise and lower this condenser. I think they all should be in the upright position when the microscopes are sitting out right now. And that's typically the most appropriate position for looking at living specimens. Sometimes when you look at other things, it helps to move that condenser up and down, and it gives you a little bit better image. So the condenser basically helps to influence and focus light that's coming up from this illuminator going through the stage. There's also an iris, and this is really hard to see. There's a little lever that sticks out right here that you can rotate back and forth. And the iris really works like the iris of your eye. And it opens and closes and lets more or less light through. Typically speaking, as you increase in magnification, you probably will need more light. Okay? If this illuminator is all the way up when you start, and the iris is all the way open, and you start looking at things under the scanning, the lowest power objective, sometimes you'll just see this, bright, you can't distinguish any detail at all. It's kind of like when light's shining through your windshield on your car, it just kind of obscures your image and ability to see. So if that happens, you can turn down the illuminator or stop down the iris a little bit. But that's an adjustment you probably will have to fiddle around with as you change power each time, and as you look at different slides as well. Let's take a look at another view of your microscope. Maybe. It's not good.
Did it freeze?
It froze. Okay, there we go. Okay, here's some other things you need to be aware of. Here are your objectives. You can see 3 of the 4 objectives. The last one is back here behind this little short one. The length of objectives is proportional to their magnification. So this is the scanning power, which is the lowest magnification. Low power. High power is back here behind the scanning. And then this is the oil immersion objective. And we'll only use this once. Okay? Anybody know what the total the magnification of the scanning power is by any chance? That magnifolds 4-fold. That is the 4x objective. Okay? The magnification of your ocular is 10-fold. So the total magnification with the scanning lens in place is 40-fold. Okay? It's a multiplicative factor. You multiply the objective magnification times the ocular magnification. Low power, the one that has a little yellow band on it, is 10x magnification. So total magnification with the low power in place would be 100. The one back here, which has a blue ring on it, I believe, is the high power, and that is 40x magnification, so 40 times 10 is 400. And then the one with the white band on it is the oil immersion objective. That is 100 times magnification. So we can get 1,000 times, 100 times 10, when the oil immersion objective is in place. But there's some other special things you have to do to use oil. We'll talk about in just a second. Here you can see a little bit better where this little lever is that pulls this particular stage clip back. And you can see how the stage slide holder right here, a slide would fit right in here if this retracted. So slide your slide in there and then let this loose, and it will nudge up against the edge of the slide. And then again, your X, Y controls you can use for moving your stage left and right, up and down. Here is your coarse focus. It's also over here. And here is your fine focus. You only should have to use your coarse focus once if you properly are setting your microscope up for use. And I'll explain that in just a second. I talked about the oculars. The oculars, there are two of them. Dr. Morton's going to talk to you at the beginning of lab about how to adjust the interpupillary distance. That is the distance between the two oculars, because some of you have eyes that are farther apart, some of you narrower. And it's really important that you adjust this interpupillary distance so that you get one field of view. You get one image as opposed to you're seeing two separate images. So you adjust those so you merge and get one image. You're really defeating your purposes if you close one eye and just look through one objective. Okay? Your eyes are going to get fatigued if you do that. So adjust this. Dr. Morton can help you to get the interpupillary distance proper so that you can then look through both objectives having both eyes open. Both oculars, sorry. If you've ever used a set of binoculars, binoculars have a little adjustment on them on one of the eyepieces that allow you to focus for differences in your two eyes. The microscope has that as well. It's called the diopter ring. And so what you do is you set up your slide so it's on low power. You bring it into focus using the coarse and fine focus, looking only through the right objective. You don't look through your left one first. Focus critically with this, looking through this one objective with your right eye. Okay? And then look through your left objective with your left eye only, and you can use this little diopter ring to independently focus the left eye. So that way both of them will be in focus, even though you might have some differences in your two eyes. Okay, the way you should always start is the microscope should be with the scanning objective in place, and the stage should be in the full upright position. The way you move the stage is to move these coarse and fine focus. Okay? So on these microscopes, the objectives are fixed, and you focus by lowering and raising the stage. There are some microscopes where the stage is fixed, and you raise and lower the objectives. Okay? So make sure the stage is always in the upright position and you're on scanning power. When you focus then, you'll begin focusing by moving the stage slightly away from the objectives. You always focus downward. Okay? That way you're not moving the stage toward the objectives. That could run the stage into the objective and break the objective lens. Okay? So full upright position and then focus downwards, move it away. When you put it on scanning first, you're going to be able to see very little detail. Really all the scanning is to kind of find your place on the slide. Use your coarse focus first to get it into general focus, and then the fine focus to make tiny adjustments. When you want to look at a higher power, which you typically will, you then just swing this low power objective into place. These microscopes are parfocal, and that means when you switch objectives, they are still in approximate focus. So when we swing this objective into place, we should not have to move the fine focus any longer. Coarse focus, sorry. We should only have to tweak it with the fine focus. So there's only one time you should be fiddling with this knob, and that's getting things focused on the scanning. Once you've seen things on the fine focus, you can shift it over again to the high power. Again, use your fine focus to bring it into critical focus. At this point, you might have to start fooling around with the illumination a little bit. Okay? So you might have to go ahead and pump up your lamp illumination a little bit maybe, or open and close the iris to let more light in. You can use a combination of those two controls to control light. But get in the habit of doing that. If you are going to use oil, and there's only one exercise where we're going to use oil, once you've looked at it on high power, turn the objective so that neither the high or the oil is in position. It's going to be kind of in between the two. Your slide will be sitting on here. The slide will not have a cover slip. And what we're going to do is we're going to put a little drop of oil, there'll be a little bottle for you, directly on top of the slide. And then you're going to rotate that objective over into place so it comes in contact with the drop of oil and the slide. So there's actually going to be a little cone of oil between the slide and the objective. And that's going to help you to get better resolution. We're not going to talk about why that's the case in this class. But really, for the oil immersion to be effective, it's got to have oil on it. Okay? When you're done, make sure that you swing this back over again so it's accessible, and then use a piece of lens paper to clean the oil off of the objective. We won't be cleaning the oil off of our slides. Okay? So make sure you go through this process with any slide you look at. Start at scanning, then move to low, then move to high, and if necessary, then move to oil. And if you do that systematically, you really shouldn't have much problem. These are very friendly microscopes. They're pretty easy to use. Keep your lenses clean. If it looks like you're getting kind of a smudgy, blurry image, there will be some lens paper at your tables, and you can use those to clean the objectives. You can also use these to clean the oculars. The oculars get kind of nasty sometimes with grease from eyebrows and things like that. In one of the oculars, there should be a little pointer. And you can actually rotate that ocular around a little bit if you want to move it to point on something, or you can move your stage. And so that's very helpful if you're asking Dr. Morton, for example, what is this? Okay, you can put the pointer on it, and then you've got a point of reference. And then this one, it's hard to see. There's a condenser focus knob over here on the side of this kind of black area on the microscope. So it's on the opposite side of the on-off switch, and here is the condenser. So when you turn that knob, this raises and lowers. The iris, which is the top of it, has a little lever that you can just move back and forth. Okay, you're going to be looking at two types of slides today. We're going to be looking at fresh mounts, and we're going to be looking at prepared slides. Prepared slides already have the material mounted on them, and they're permanently covered by a little glass cover slip. So the epithelial tissue, I guess, is a prepared mount. Other mounts you're going to make yourself. On one of them, we're going to put a little drop of water, and then you're going to take a toothpick and do a few little tooth scrapings. Because you have bacteria all inside your mouth. And we're going to emulsify that a little bit in this drop of water. And then we're going to add the methylene blue to that? Yeah. So we're going to add this stain called methylene blue. And we don't have to put a cover slip on it, do we? No. So it's going to dry first. It's going to dry first. The drop of water will dry after you put the methylene blue. So you'll put this on the stage without a cover slip, and then when it comes time to using the oil lens, you'll put your drop of oil directly on top of that dry piece of tooth material that's been stained. Pretty much everything else, I believe, is going to be a wet mount. And a wet mount is made by putting a little drop of water in the center of the slide, or taking a drop of culture that's liquid and putting it on the slide. And then you're going to lay one of these little transparent glass cover slips on top of it. It's important to do that properly. If this is my slide, and this is my cover slip, not exactly the right size, but if I've got a drop of water here, and I just take that cover slip and do like that, it's going to trap some air bubbles underneath. And I can pretty much guarantee that somebody at some point in time will ask Dr. Morton what this is, and it will be an air bubble. In fact, sometimes we'll actually have you make our bubble slides so you can see them. But air bubbles are usually nice and round, and one of the things that characterizes them is you focus up and down. Oftentimes one edge of the bubble will be blacker than the other, and the position of that will move as you focus up and down. And so be aware of air bubbles. If you want to minimize the chance of getting air bubbles, drop of liquid in the center of your slide, touch the cover slip to the edge of the drop of water, and then gently lower it this way, and that's going to tend to push the air out. On some of these slides, we will ask you to put a little drop of sticky material called detain, or sometimes it's called proto-slow, onto the drop of water. And that just makes a more viscous solution, and it slows down some of these organisms that are very mobile. Paramecium, for example, really swim pretty fast. And so in some situations, that's going to be necessary. What am I forgetting?
The stereoscope's
Okay. Well, I should have taken a picture of a stereoscope. Okay, we're going to use a different kind of microscope to look at hydra, called a stereo microscope. And it's set up a little bit differently. You're going to put little culture dishes right on the stage. In this one, the stage is fixed, and as you turn the coarse focus, it raises and lowers the big objective lens to give you your magnification adjustment. On the top is a little rotating knob that allows you to increase or decrease magnification. Those stereoscopes don't have nearly the magnifying power that these do, these compound microscopes, but they really are much more effective in seeing things kind of three-dimensionally, and hydra are fairly big, and so we'll be using the stereoscope for those. When you're getting your cultures out of the containers, many times they tend to settle toward the bottom. And there'll usually be a little plastic pipette in there that you'll be asked to use to take some of that culture out. If you push on that bulb first, while the pipette is immersed in the liquid, then that's going to push air out of the bulb, and it's going to push everything away from the pipette tip. And then when you release, you'll be sucking up pretty much clear water. So what you should do is pull that pipette out, press on the bulb while it's out of the container, hold that bulb, put it in the container, and then release. And then you're not creating this convection current that pushes everything away. When you do use the detain or the proto-slow, sometimes that tends to push things out towards the edge of the drop you're looking at. So a lot of times you have to focus kind of around the edge of that drop to really see something. What
else? Whatever's comes to mind, I can take care of in class.
Okay. We thought we would show you real quickly just a few images of what you're going to be looking at. And actually these images will be on the computers in their lab, and probably what Dr. Morton will do is have you log on and just kind of have some of those available. Because sometimes it's helpful to be able to see what you're supposed to be looking at, so you've got an idea. This is a nice image, nice colorized image of some bacteria that you typically find in your mouth. So these are epithelial cells, and then these are some of the bacteria types that are found on there. This is actually a stain preparation from a student's mouth, and shows some of the bacteria that you'll find in that after the staining process. So you will see something that looks like this when you do your tooth picks. Here's an amoeba. Amoeba, I think, are very fascinating single-celled organisms. These guys are really, really hard to see unless you really, really cut down the light. So one of the things that you really have to do with the amoeba is move that iris lever back and forth and almost stop it down to the point where it's almost entirely closed. Because the contrast between these relatively colorless cells and their background is really hard to differentiate. And so if we cut down the light, we get a little bit more contrast. I think Dr. Morton is going to having you add a dye to this called neutral red. And neutral red is a dye that can be used as a vital stain. Living organisms take it up. and it will be taken up into some of these little vesicles and vacuoles here, and they will stain red very quickly. So that's kind of a cool thing. This is a filamentous prokaryotic organism that is photosynthetic called annabina. So each one of these little cells right here is an individual prokaryotic cell. So that's one of the prokaryotes we'll look at. Here is paramecium. Again, unstained, so you probably will have to stop down the light. You can see kind of here the little individual cilia that surround the outside of this cell, and they meet back and forth so this thing can really motor and move. And you can see some fairly large vacuoles in this. It's hard to see the nucleus on this one, but oftentimes the nucleus is pretty prominent. Protococcus is a little green algae that tends to form small colonies. This is photosynthetic, obviously, so you can see chloroplasts in these cells, but these are the individual cells, and they typically will group together to form small colonies. Cenodesmus is another single-celled colonial organism, typically 4 cells stuck together. Little spines that stick out at the end of these 2 cells, and you can see chloroplasts on those. Volvox is a very interesting colonial organism. This is made up of a huge number of cells, so these are the individual cells right here. They all form this big spherical colony. And then these are small daughter colonies that are forming within the Volvox. And oftentimes these will break open and release these daughter colonies. These cells are flagellated, they have little flagella on them, and so the whole colony will spin and rotate. And it's really pretty impressive. Those are large. You can almost see those macroscopically with your eye if you look at the culture. So these, 4x or 10x would probably be appropriate. This is Hydra. This is a multicellular organism. These are its tentacles that they can use to capture prey. And then this is kind of the mouth area, so they grab little things like water fleas, and they can paralyze them. They've got little stinging cells on their tentacles, and then they can kind of stuff the food down their gullet. And their body is basically just a big sac, a big digestive sac. But they're kind of cool to see too. This is the one we're going to use the stereo microscope to look at instead of the countdown microscope. Intestinal tissue. This is a cross-section of the intestine. So this would be the interior of the intestine, of the tube, called the lumen. And then these are the cells that separate the intestine from the next layer of tissue, which is oftentimes really heavily filled with blood capillaries and things. So food that's being digested will eventually have to be absorbed and cross these cells, cross these cells, cross these cells, and eventually get into the circulatory system. You can see these little foldings of the intestine to increase surface area. You may know what those are called? Those are called villi, V-I-L-I. And then as you get into higher magnifications of one of these villi, you can actually see that the individual cells, that their membrane is indented. And that forms structures called microvilli. So lots of adaptations for increasing surface area. So here is a villus, and you can start to see individual cells right here. Here's the nucleus of the cell. There are a couple of kinds of cells that are found in this villus. And these cells right here that have this kind of empty looking area here are called goblet cells. And the goblet cells actually secrete mucousy-like material that then is secreted out into the lumen of the intestine. These other ones that don't have that little whitish area are the columnar epithelial cells. So you'll be able to see those. This shows you the villi and then some of the muscles that are associated with the intestine. So you'll look at some of these muscle layers a little bit. So you will be going from single-celled prokaryotes all the way to tissues of living organisms that consist of many cells that are all eukaryotic, and several of which are differentiated into different cell types. Muscle type tissue, more secretory tissue up here. So Dr. Morton will keep you busy for the most part for the lab period. But that'll give you an idea of kind of what you're looking for. Okay?
Yep.
Do we have any questions? Okay? We will hand out your quiz to you, and if when you're done, if you would put them up here again by lab section, and